we've been focusing on, oh, DNA controlling life. The epigenetic story actually shifts our focus to looking at how environmental signals are controlling the DNA in our lives. So all of a sudden, it changes our belief system. It says, no, DNA is not self-actualizing. DNA expression is related to our response to the environment. So I say, okay, let's go back to the cells in tissue culture. And as I said, as I put the tissue culture cells in the plate and then feed them with culture medium, it is the culture medium that determines the fate of the cells. And I go, well, here's an interesting point. Well, we've been talking about cells in a petri dish. It's really no different than cells in your body. Remember, your body is comprised of 50 trillion cells. That, in fact, you can perceive of your body as a skin-covered petri dish, and within that dish are 50 trillion cells. So a human body is a petri dish. And I say, yeah. And just like our study in the laboratory, adding culture medium to the cells, well, in the body, the culture medium is the blood. So all of a sudden we say, well, it is no difference if the cell is in the plastic dish or the cell is in the skin-covered dish, because the cell is still going to respond to the culture medium environment. And in the human body, that culture medium is blood. I say, well, that becomes important because then the chemical composition of the blood is actually controlling the fate of the cells. And I say, well, there are two aspects to what is in the blood that is controlling biology. Number one is just the basic nutritional elements. So in culture medium, there's nutrition. And this is significant because it says when we make the culture medium called blood in our human body, it also is derived from nutrition. So one of the big factors affecting our health is, are we taking in proper nutrition that will support the health of our bodies, or are we taking in toxic elements as produced in industrial farming? Because these toxic elements will interfere with the growth medium, and in turn, via epigenetics, the toxic food that we eat will alter our genetic expression and can contribute to our health. Well, in addition to nutrients, blood contains cellular signals. I go, cellular signals? I say, yeah. Emotional chemistry, hormones, growth factors. So blood contains nutrition and blood contains information. And the cellular signals come from the nervous system. So to understand how we create the blood composition, recognize this. We live in an environment external to our body as well as we have an environment internal in our body. And I say, well, how does the cell know what the environment has in it? And the answer is this. The function of the nervous system in the brain is to read the composition of the environment in which we are living. So the function of the brain is perception, what is going on in the world. And then the brain will translate that perception into signals to coordinate the body's response to that environment. So the signals from the brain are passed via the nervous system and the circulatory system to the cells of the body. These signals in the blood are the factors that are then controlling epigenetics, and so brain-mediated signals control behavior and gene activity by being transmitted through the nervous system and blood to the target cells. When we get to the level of the human, it's more than just the environment that is controlling behavior and genes. When we get to the human, we start to recognize that there is a mind. And when we put the mind into the scheme, it changes the flow of information for this reason. The brain picks up the signals from the environment, but the mind does an interpretation of those signals, and that interpretation is translated into the chemistry released by the brain. So the culture medium components controlling biology are not necessarily directly related to the real world. The culture medium contains information that is directly related to our perception of the world. If we have a misperception of what's going on in our environment, then you realize this. The signals that our cells receive internally is not the signal of what's really going on in the world. And so the cells don't see the real world. They only see our interpretation. That interpretation translated by the brain turned into chemicals that are released into the body, causing the composition of the culture medium that influences cells. So we now have to recognize that the environment that we are living in is really creating the chemistry that is controlling our biology. To simplify an understanding that, in this cartoon, what we can see is an image of a brain. And in one hand of that brain, there's a flask of culture medium. In the other hand, the brain is feeding that culture medium to cells in a dish. And I go, okay, that cartoon represents the fact is this. The brain is creating the blood, the culture medium, and that blood is then used to control the fate of the cells. So I say, okay, well, how does it work? And I go like this. When we have a perception, that perception is translated by the brain into the chemistry used in the culture medium. So I can say, when you open your eyes, let's say, and you see someone you love, the brain interprets love and translates it into chemistry such as providing the dopamine as a result of experiencing love, which is the pleasure we experience in love. In addition, when you're in love, the brain will release oxytocin into the culture medium. And the oxytocin is to bond you to that source of love. Then the other chemicals that come out of the brain include vasopressin. So when you're in love, vasopressin is a chemical that makes you more attractive, so that your partner, your lover, will stay with you. And lastly, brain activity in response to love releases growth hormone. And growth hormone, as the name says, is a chemical that supports the growth and health of the cells in your body. So a simple point is this. When you're in love, the chemistry released by the brain into the culture medium is chemistry that enhances your health, your vitality, and makes you much more attractive in the world in which we live. And this is a wonderful experience and a consequence directly due to the perception of love. But in contrast, if you open your eyes and you see something that scares you, there's a completely different set of chemicals released into the blood. No longer are those vital chemistries such as growth hormone and dopamine released when you see something in your environment that causes you to be afraid. Because in that situation, the cell will begin to release stress hormones and inflammatory agents that put your body into a protection mode to protect you from the threats on the outside. So if you put the chemistry of fear into the culture medium, it alters the expression of the cells. In fact, in a laboratory situation, if I create culture medium and put those stress chemicals into that culture medium and feed my cells, guess what? Within minutes, the cells start to get sick, and then shortly after, they begin to die, which really supports the known belief that stress can cause death. And in fact, stress is the most important factor that apparently is influencing the health on this planet. So we can come back and say, well, the function of the mind is to create coherence between what we believe and the reality that we experience. If you believe there's a threat, the signals of threat released into the blood will prepare the cells to engage in a protection response. But the problem is this. Maybe there was no real threat. The cells don't know whether it's real or not because the cells only respond to the chemicals. They don't see the real environment. So all of a sudden I say, then if we believe we're under stress, we manifest stress in our physiology, even though the environment in which we live is not really promoting that. So all of a sudden we start to see that our thoughts, whether they're right or they're wrong, are actually changing our biology. Now, this idea of thoughts controlling our life is not new. 2,500 years ago, the Buddha said, what we think, we become. And in fact, that is exactly the story of epigenetics today. Epigenetics is saying what you're thinking is translated into chemistry, which will determine what you will become. So, 2,500 years later, Buddha is right, but now we know the mechanism. Signal plus protein equals behavior. Now I say, when the behavior is not supporting us, then we can say, we are expressing a dis-ease. Now, in this equation, I say, well, what can be responsible? What can cause a disease? The beautiful part about it is there are only two things on the other side of the equation. It's either the protein or the signal can be considered the cause of disease. So I say, well, yeah, proteins can cause disease because defective proteins lead to defective functions, which then are expressed as disease. So the question is, how much of our disease is actually controlled by defective proteins? And the most amazing fact is this, that flawed genes cause less than 1% of disease. So when we go back to our chart and I say, proteins can contribute to disease, and I say, well, they can contribute because defective proteins as a result of mutations or alterations of our genome then will mislead the fate of the cells, and in that case, cells out of harmony with the world express disease. Now we're down to a very simple understanding. If only 1% of disease is related to proteins, going back to the chart says, well, then the other cause of disease must be some alteration in the signal. So I say, the signal can cause up to 90% or more of disease that we experience on the planet. I say, well, how can the signal interfere with health? And I say, number one, if you experience trauma, let's say you're in an accident and you twist your spine and you interfere with the flow of information from the nervous system to the cells, then the alteration of that interference of communication could lead to a misunderstanding by the responding cells. So what's really going on because the signals are altered? So trauma can cause disease by interfering with the propagation of the signals from the brain to the cells. Number two, if we put toxic elements into our body, such as the toxins that we acquire by eating industrial farm foods, toxic chemicals can engage in the signal pathway, but if they do, they don't promote a normal signal propagation. Toxic chemistry can distort the signal. So relevance is this. If the brain is sending a signal to control the cells and there's toxic chemistry in the pathway, then the signal that reaches the cells is by definition altered, and therefore the behavior of the altered cells can lead to disease. Thirdly, and perhaps the most important cause of disease influencing the signal, is really nothing wrong with the body at all. But what it represents is this, the influences of our thoughts. Our thoughts are what are translated into the chemistry. Now, why is this relevant? If we have a disease due to thought, it's not because the body is defective, it's because the signal is inappropriate. And this is really important because it is recognized now that our consciousness is the primary problem in the issues of health on this planet, especially as they relate to stress. And that this issue is nothing organically wrong with the body. It's just that we're sending inappropriate signals at an inappropriate time, which leads to inappropriate behavior, which we call disease. So I look over, I say, look, there are only three causes of disease that interfere with the signal. One, trauma, and trauma represents a physical impairment. Two, toxins, which represent a chemical impairment. And three, thought, which introduces an emotional impairment in the system. And collectively, all of these things result in what we've referred to as stress. So stress is the primary cause of illness, with proteins attributing only about 1% toward that pathology. And I say, well, is this really the understanding? I go, the American Psychological Association, in an article entitled Stress, How Does Stress Affect Us?, comes to the conclusion that from 75 to 90% of all doctor visits are actually due to the influence of stress. And all of a sudden I say, yeah, but then that means 75 to 90% of all doctor visits can be eliminated if we understood how to handle that stress. So I say, well, first of all, before you go deep into stress, we must recognize there are two kinds of stress. One is called eustress. Well, that's good stress. You say, there's a good stress? I go, yeah. If you play sports, for example, and you're out on the playing field and you're running and jumping and moving around, by definition, you're stressing the physiology of the body. But it's not a bad stress because the resolution of this stress is the body's ability to strengthen itself, make itself be able to carry out those athletic needs. So yes, I'm stressing the body when I'm playing a sport, but it's not affecting me in a negative way. It's actually affecting me in a positive way because it's inducing more health from my cells to accommodate my needs. The issue we really want to deal with is called distress. And the significance of that is a stress that promotes a negative environment. I say, well, what does distress mean? And it's very simple. Distress is a result of anything that gets in the way of your destination. Whether your destination is a career move, or whether your destination is a relationship, or whether your destination is trying to take care of your health or your financial background, anything that interferes with those, by definition, is a source of stress. So distress is the bad stress that interferes with health. And I say, well, how does it interfere with health? And number one is simply this. When you put cells in a petri dish, I can create an experiment where I create two sets of dishes. In one set of dishes, I put nutrients in front of the cells to support their growth. In the second set of dishes, I put a toxic element that threatens the cell's life. I put these culture dishes back into the incubator, wait for a while, come back, take the dishes out, and then I look, where are the cells in each of these two dishes? Well, number one, when you put nutrients that support the life of the cell in the dish, the cells move to the nutrients to take in the nutrients, assimilate them, and put them in their body. But in the petri dish with the toxic elements, the cells don't move to the toxic element. In fact, they move the opposite way. The cells move away from the signal, and in this case, in the form of protection, they also close themselves down to wall off the threat. So we have two different behaviors based on the signals. A positive signal causes the cell to move to the signal, open its arms, and take in, assimilate the positive signal. A negative signal causes cells to move backwards away from the signal and close themselves down in protection. The point is this: cells cannot move forwards and backwards at the same time. Cells cannot be open and closed at the same time. The net result is a very simple conclusion. Cells can either be in growth, moving to the signals and assimilating, or cells can be in protection, moving away from the signal and walling themselves off. And the important part about this is that these behaviors are mutually exclusive. A cell cannot be in growth and protection at the same time. So, when we look at this picture and understand it, then what we can recognize is a simple fact. Protection mechanisms wall off the body and stop growth. I go, well, that sounds like a very bad situation. Why would we do that? Why would we shut down our growth mechanisms when we're under stress? That might be the time we need to activate those mechanisms. The answer is no, for a very simple reason. Historically, in evolution, go back to primitive people. And I say, well, tell me about their lives. And I go, well, very simple lives, looking for food, hanging out in the trees, and just enjoying the Garden of Eden at the time. But then I say, occasionally, a saber-toothed tiger shows up. And I go, well, that means the human has to protect itself. And I say, well, what's the human going to do? Engage in fight or flight. This is the engagement offered by the hormones of the adrenal system. I say, yeah, but what does that mean? I say, well, imagine you're running away from a saber-toothed tiger, and I say, how much energy do you want to allocate to your fight or flight mechanism to escape the tiger? And the point is logically very simple. All the energy that I can make available, I want to have available to escape the tiger. So I say, yeah, but the energy in the body is being used to do many different things, such as growth and maintenance of the body, taking care of it, growth functions, and energy is actually used for the immune system. And it uses a lot of energy, that immune system, only because, as you well know, when you're sick, most people don't have enough energy to even get out of bed. So the immune system uses a lot of energy, and growth uses energy. So I say, you're being chased by a saber-toothed tiger, how would you split up the energy? And I would say, stop the energy for the growth mechanism. Why? I don't need to grow right now. I need to escape and save my life. I don't escape. Growth is irrelevant. The same thing applies to the immune system. If I'm having a bacterial infection and I'm being chased by a saber-toothed tiger, what would be the benefit of funding the immune system with energy? Because if the tiger catches you, that bacterial infection is not your problem anymore. It might even be the problem of the tiger. So the point about it is simply this. In a state of threat, stress hormones are released from the pituitary master gland to control the protection response in our body. The stress hormones do two things. They shut down all mechanisms that utilize energy that are not necessary from escaping from the tiger. So growth and the immune system are inhibited under the influence of stress hormones. When the system evolved 1,000,000 years ago, the only stresses that people had were that occasional saber-toothed tiger. So there will only be short intervals of using a protection response, just long enough to escape from the tiger. When the individual had escaped the tiger, they were no longer needing a protection response. That stress system would shut down and the system would return back to normal growth. But that was 1,000,000 years ago. Today, the stress that we face in our world is not little short bursts, but actually 24/7, 365. Every day, based on what's going on in the world, financially, health-wise, whatever, stress of that information is released in our body in the form of stress hormones, and they end up shutting down the growth and the immune system. And I go, well, that's not the original intention, a short burst of protection, because chronic protection leads to chronic problems because of its ability to shut down the maintenance of the body and the immune system. So all of a sudden you realize, why do we have so much illness in regard to the world we live in? And the answer is not because of our genetics and our biology, but because of the level of stress that the citizens have to deal with from day-to-day life. And that if we could eliminate that stress, then what we can do is actually recover our health again. So our health is not due to a defect in our body. Our health is just due to a mechanism that is out of control because of the amount of stress that we are creating in the world. So, two consequences of stress. A, when the stress hormones are released into the body, they start to close the blood vessels in the gut, and that stops the flow of blood to the gut. The blood, of course, is the source of energy. And I say, well, why would I stop the flow of the blood vessels in the gut when I'm being chased by a tiger? And the answer is simple. If you close down these blood vessels, it pushes the blood to the arms and legs, which are going to be used in the escape process. So we're just allocating energy to the somatic system to escape the threat. But if the threat is prolonged, then you have to realize those blood vessels shutting down the gut functions will actually inhibit the maintenance and caretaking of the body, and prolonged stress will have detrimental effects on our fate in regard to our health and our ability to survive. So that's aspect number one, shutting down the growth. Aspect number two I also mentioned was this. The immune system uses a tremendous amount of energy. So the idea of an internal immune problem and an external saber-toothed tiger easily says, forget the immune system at this moment. Let's put the energy into running away from the tiger. And I say, yes, that is one of the functions of stress hormones. Stress hormones shut down the function of the immune system. It is so effective that medical doctors give patients that are going to receive a transplant of an organ, they give those patients stress hormones before the transplant. This reason is quite simple. The ability of the stress hormones to inhibit the immune system that actually protects the recipient of the new organ from having their own immune system reject the graft. And this supports the life of the transplant. So the effectiveness of inhibiting the immune system is so much, it becomes a therapeutic tool in organ transplant. So I say, okay, our life is now under threat and our hormones in our body are shutting down growth, and the immune system, there's only one consequence of that, and that is a breakdown of the system and infection. And this is really the primary cause of the illnesses that we have on this planet. And one other aspect, which I call adding insult to injury about stress, is this. The prefrontal cortex portion of the brain, which is just behind our forehead, is the site of our conscious mind. And interestingly enough, while the conscious mind is creative, it's a very slow processor. The hindbrain has reflex behaviors which are instantaneously fast responses. When they're under stress, the adrenal hormones squeeze the blood vessels in the forebrain just as they squeeze the blood vessels in the gut. I say, yeah, but when you squeeze the blood vessels in the forebrain, it forces the blood to the hindbrain. That, in other words, the blood energy is not being used in the forebrain under stress, but is being used to engage the reflex behavior of the hindbrain, the much more effective mechanism to save us in a very scary, stress-inducing situation. I go, relevance is this. Forebrain is consciousness, hindbrain is reflex behavior. And when we're under stress, the hormones shut down our consciousness and cause us to behave in a reactive, reflexive mode. That makes us less intelligent. The net result is there are 3 consequences to elevated stress hormones. 1, shut down the growth using blood vessel closure in the gut to stop the functions. 2, shut down the immune system because stress hormones inhibit the inflammatory role of the immune system. And 3, stress hormones make us less intelligent because it shuts down consciousness and engages reflex behavior. You put all these 3 elements together, and then all of a sudden you have a profound understanding of the cause of disease. The 2 basic ones, physically, not supporting the growth and maintenance of the system, and inhibiting the immune system, lead to a physical breakdown of our biology. And that is why up to 90% of doctor visits are primarily due to stress. In addition to the immediate influence of stress, stress has a long-term interference with our vitality and the survival of our biological system. And it influences this effect via something called telomeres. And you say, well, what are telomeres? And I say, understand that a DNA molecule is a double helix, as Watson and Crick revealed. That it will stay functional as long as the molecule stays in its helical configuration. But if you start to unfold the double helix into single strands, this opens the DNA to a breakdown process where you can destroy the gene programs. So you don't want the helix to unfold. I said, well, how do you prevent it from unfolding? Well, to give you an idea, I'll show you a picture of a shoelace. And at the end of the shoelace, there's a special little plastic cap that holds the threads of the shoelace together. It's called an aglet. And I say, well, great. The aglet holds the filaments of the shoelace together and makes it easier for us to stick the shoelace through the little holes into the shoe so we can tie our shoes. I go, yeah, but if the aglet comes off, the end of the shoelace frays, and then now you start to ruin the function of that shoelace because you can't get it through that little tiny hole anymore. And I go, in a parallel understanding, the DNA is the wound up version of the double helix, and to prevent it from unfolding, there's the equivalent of an aglet called a telomere. That holds the ends of the DNA together to prevent them from unfolding, okay? If the telomere is gone or breaks down, then the DNA will start to unfold, and that would have a negative aspect on our health because it can cause many of the diseases that we experience, such as aging, depression, and other physiological things like cancer, etc. So the telomeres are playing a very important role. And yet what we've looked at is that they appear to be nothing more than the equivalent of an aglet to hold the DNA together. I go, no, there's more to the telomere than that. And that is this. Why have the telomere? Yes, to hold the DNA double helix together, but there's another more important reason. These telomeres, as you can see in the picture of an isolated X and Y chromosome, these telomeres stained red are at the end of the DNA where we would expect to find them. And I go, but what do they represent? They're extensions of the DNA, but they don't program anything. So they're not genes, they're just nonsensical in one aspect, extensions of DNA, no gene program. I say, so yes, they're there to hold the DNA double helix together. And I go, yeah, but there's another important reason for telomeres, and that is this. In copying the DNA, you have to split the DNA apart into single strands. And when you do that, an enzyme called a DNA polymerase will land on a single strand of DNA, and like a train going down the tracks, as the polymerase enzyme moves down the length of the DNA. Behind in its wake, it leaves a copy of the DNA. So imagine a train going down the track, and right behind the train, there's a reproduction of the track. So all of a sudden, now I have 2 tracks where I had 1. This is how you copy the DNA. And I go, well, this is really great, but here's where the problem comes in. When that enzyme reaches the end of the DNA, and it's sitting on the DNA before it falls off, it cannot copy the piece of DNA upon which it is sitting. So I create a copy of the DNA that I'm on with the enzyme, but when the enzyme gets to the end of the DNA strand, it cannot copy that last piece. And I say, well, what's the consequence? I say, the new copy of the DNA is shorter than the original copy. I go, oh, okay. I say, yeah, but then the next time it divides, the same thing will occur. So then the next copy of that DNA is still going to be shorter than the previous copy. Every time you copy the DNA, you lose a piece of the telomere because it cannot be copied by the enzyme, because the enzyme is sitting on top of it and needs to pass over it. And if it can't pass over it, it can't leave that DNA behind. So the net result is this. Telomeres, yes, provide for holding the DNA together to prevent it from fraying and then causing its breakdown. But as, or more importantly, the telomere provides a platform that allows a large number of reproductions of the DNA. Considering that every reproduction is shorter than the previous version, it will say what? At some point after so many cell divisions, the DNA called the telomere will be lost because it's shortened every time previously. And I said, well, what happens if you copy the DNA and the telomere is not there? And I say, oh, well, the next copy is going to be short again. But what's important is what you're shorting is not an extra piece of DNA. You're shorting a piece of a gene program that makes a protein vital to our biology. So when the telomere runs out, the next copying of the DNA will interfere with the genetic code and alter the expression of the organism. It was then understood that telomeres determine longevity. As long as you have telomere, you can reproduce the DNA, but if you divide the cell too frequently, the telomere disappears, and the next version, shorter version, will be shorter because you're cutting off a portion of a gene program. And that interferes with our biology and can cause aging, disease, and depression. So I say, oh, when this was originally understood, a scientist by the name of Leonard Hayflick in the 1960s said, well, if every division makes it shorter, then there's only a finite number of cell divisions a cell can have. When it reaches that number of divisions, after that, disease will result, and then death will occur. So it said, oh, our life, the length of our life, is determined by the length of the telomere. The shorter the telomere, the closer you are to aging yourself out of existence, because when the telomere is gone, that's when the disease enters into the picture. So the original thing said, oh, all humans have a fixed lifespan because they have a fixed DNA extension called a telomere. And that all of a sudden led to counting how many cell divisions a cell will be able to experience before losing its telomere, and then put that into context of how many cell divisions we experience in our life. And it says, well, a human may have a life of anywhere from 80 to 90 to 100 years based on the amount of telomeres that are maintained in their body. Well, that said, oh my God, our lifetime is fixed. However, a woman by the name of Blackburn identified an enzyme called telomerase. Telomerase is an enzyme that extends the telomeres. I said, oh my goodness, if there's an enzyme that extends the telomeres, then the concept of a fixed life is totally wrong. Because as long as I can extend the telomeres, I can continue to divide the cells and maintain my life. So there is no limitation on how old we are, because we can use the extended telomeres to extend our lives. This then leaves us to a final understanding about the influence of telomeres in our lives. And it really comes down to this aspect, and it says this. There's the enzyme that can influence the production of more telomeres. If we activate the enzyme, we can extend our life and our health. If we don't activate the enzyme, our life will be shorter and will be influenced or affected by disease much more easily. So I say, okay, there are things that will enhance the telomerase, which will increase our life and our health. And there are factors in our life that can stop the telomerase and in the end cause us to have premature death and disease along with that. So I say, well, what are the conditions that are responsible for, let's say, inhibiting the telomerase that cause us to have the diseases that we have on this planet? And the answer to that is, here is a list of the following factors that will stop the telomerase activity, shorten your life, and cause you to have more disease. And these factors include stressful prenatal development. And the significance of that is, if you're stressing the cells in the very early part of life, you're promoting more cell divisions very early that will then use up the telomeres, and because of the stresses, you're not going to extend them. So the result is stressful prenatal development will shorten the duration of an individual's life. Childhood abuse will do the same thing. Now, people think childhood abuse is physical abuse, hitting your child. And I go, well, that is childhood abuse, but what you must understand is this: childhood abuse can also result from verbal abuse. So they say, no, I didn't hit my child, I just yelled at him. I say, no difference. Yelling at the child, hitting the child have the exact same effect on the telomeres because they inhibit the telomerase activity. Domestic violence, another factor that will shut down the enzyme. Post-traumatic stress disorder is also in that community of inhibitors of telomerase. Two last things to consider are this: nutritional deficiency. And the reason why that interferes with the telomerase is that we don't have the metabolic support to provide for telomerase activity. And let me just add this as what I think is the most important of the influences that shut down telomerase and lead us to disease, and that is a loss or lack of love. Love feeds the system, encourages health and growth. As I said, the experience of love releases growth hormone into the body, and in that environment we can create more health. But when there's no love, the telomerase activity will shorten itself. All of these things between violence, stress, lack of love say that we're not enjoying our lives. And the response to that is then our biology says, oh, okay, well then we can shorten it right away because we can use up the telomerase and get out of this lousy situation. I say, yeah, but look at the opposite consequences, the opposite things that result in enhancing the telomerase activity. Number one, good nutrition, and especially including omegas. Omega lipids are vital to supporting telomerase activity. Exercise, as I said, that's eustress. That's the thing that builds up the biology, and in building up the biology, again, that's another activator of telomerase. Love and self-love increase health and are feedback to the system. It says, I love my life. Just literally what you're telling yourselves is, I want more life. Love enhances telomerase to give us a greater opportunity to experience love on this planet. Along with love is positive outlook. If you see the future is bright, you're telling yourselves, I want to be here for the future. If you have a very negative outlook, what you're telling yourselves is, I'm afraid to be here and I don't want to be here because it's going to be very stressful, whatever it is, and you'll shut down the enzyme. So love, positive outlook, happiness and gratitude are all thoughts of the brain translated into chemistry to engage telomerase, because those are the thoughts that say, I love my existence on this planet, I want to be healthy and I want to maintain myself, and as a result, these thoughts and actions cause the telomerase activity to be increased, which ends up extending our life. And lastly, and most importantly, is that being in service activates telomerase. What does that mean? Are you a caregiver? Are you providing health for somebody? Are you providing something that other people need to stay alive? You start to get a conscious understanding that says, I am responsible for taking care of these things. I don't have the time to die early. I need to stay healthy longer because I have these jobs. So being in service is actually promoting telomerase activity and increasing your lifespan. Okay? And let's consider the opposite, which usually affects people after they retire. I'm retired. I have nothing left to do. What does the feedback of that thought have to do with our biology? Simple. When we have no function or reason to be here, we inhibit the telomerase because the telomerase was extending our life, and our thoughts say, we don't need any more extension. We're finished. I'm retired. I have nothing left to do. And I say, just that thought alone can stop the telomerase activity. And that result causes us to shorten our lifespan and engage in more sickness as well, because the loss of telomeres are one of the factors that increase the problems in our biological system. The summary is this: our thoughts are translated into chemistry that shape our body. The shape of our body is complement to the thoughts. Positive thoughts give us a positive, healthy biology. Negative thoughts can actually introduce any disease and death. That these thoughts control our biology on a day-to-day basis and interfere with our metabolic and physiological systems if they're not supporting us. And then I go, but these thoughts and behaviors also influence our longevity, because these thoughts can alter the activity of telomerase enzymes. If we stop the activity of the enzyme, we are shortening our lifespan and increasing the chances that we will have disease. In contrast, if we engage in those behaviors, such as positive thinking, being in service, being in love, those are things that engage the telomerase enzymes, and these activities then prolong our life and make us healthier. Conclusion is very simple. We're not victims of our genetics and our heredity. Epigenetics reveals how these thoughts that we've been talking about influence the genetics and the behavior of our cells. Therefore, what we have to understand, and what we must also help others with, is that our health is not out of our control. Our health is directly under our control. The control of our perceptions, our beliefs, and attitudes actually is what will control the stresses. And the stresses are the things that cause the disease. If you stop stress, you can stay in health. And as a mission for healers around the world is to get the stresses out of the patient. So once the stress is removed, the system is able to rebuild itself using the chemistry of the brain to support growth and increasing the activity of telomerase activity, which supports a healthier, long-living biological system.